Welcome back. In the last video, Unit 4.1, or set of videos, we looked at the methodologies specific to cognitive psychology, where we compared the information processing approach versus a newer approach, the cognitive science approach. Now, one of the important aspects of the cognitive science approach is its multidisciplinary nature. Part of it is including neuroscience methodologies as one way to um, understand human mind. In this particular unit 4.2, we're going to talk about this particular uh, discipline and approach, in particular the cognitive neuroscience methodologies and the different measures we use under this umbrella to understand better about human mind and how it works. And that's what we'll do in this video. Okay, in this particular unit, 4.2 neurocognition, the outcomes are first we'll talk about the basics of neurocognition, then we'll talk about some um, principles specific to neurocognition, then we'll talk about few most important methodologies coming from this discipline, and we'll end this with this outcome of talking about nervous system in general. Again, there are a set of videos following this video which gives in detail um, the the structures and the functions associated with the different structures of the human brain. So I strongly suggest you also watch those videos if possible. So let's start with the basics of neurocognition. So, what is neurocognition? Well, neurocognition is both cognition and neurological aspects of it. In other words, we'll study the mental processes, but we'll also gather information specific to the brain's structures involved in it, both physiological, chemical, and things like those. Why? Well, it gives only additional information, right? Remember, the cognitive sciences approach main point is that with multiple perspectives or multiple methodologies, we get multiple kinds of information about human mind, which would help us get a bigger picture and a better understanding of the workings of the human mind. So, why neurocognition? Well, it gives us more information. Now, we're going to talk about some basic logic of neurocognition by looking at some specific commonly used um, reasoning to make inferences about mental processes based specifically on brain damaged patients, either because of an accident or because of an illness, and we'll talk about that. So the logic of neurocognition in terms of using brain injured patients is um, trying to see um, how closely related or separable two mental processes are. Just assume that you have patient one who has region one um, injured. So a part of the brain, say region one, is injured in one patient, and you see that this particular patient has a deficit in a mental process A, but there is no deficit in mental process B. If you have another patient who has injury elsewhere, who has a deficit in mental process B, but there is no injury in mental process A or deficit in mental process A, now you can make inferences about the relationship between these two mental processes, how associated these two are, or how separate these two are. And we'll try to understand, make sense of three specific terms here, and which we'll talk about in the next few slides: association, dissociation, and double dissociation. So association is when both mental processes A and B are disrupted. In other words, in every instance of mental process A disruption, imagine there is a patient with brain injury and mental process A is disrupted, but there is also mental process B disrupted all the time. In other words, these two mental processes are closely associated. Maybe they are sharing the same region in the brain, or they are inseparably dependent on each other, meaning that these two mental processes are associated. So association is when you only get instances of patients with disruption to both mental processes. Dissociation is when there is a disruption to of only one particular mental process every time there is a brain injured case. In other words, in every brain injured patient in the that has a disruption in a particular mental process, there is definitely no disruption in other process. So all the cases so far have been um, patients with deficit in mental process A, but not mental process B. You have another patient who also has deficit in mental process A, but no mental process B. There isn't a single case where there is a disruption in mental process B and there is no disruption in mental process A. So dissociation is when there is a disruption of only one mental process and there isn't a single case of opposite, which is a disruption of B only. So this shows that there is some level of dissociation, but there is not complete dissociation. In other words, maybe one mental process is dependent on the other mental process. So when one is disrupted, the other one is also disrupted. And you cannot imagine a disruption of the other without the first one. Again, it will more makes more sense when we look at the TV analogy and other stuff, and also another example. But I just wanted to give you an idea that association is inseparability of two mental processes. Dissociation is um, mild separability. Again, just because there is a dissociation doesn't mean that there couldn't be double dissociation, and which we'll talk about in the next slide. So we've looked at association and dissociation. Now, dissociation and double dissociation, what we'll talk about. Now, dissociation is also referred to as single dissociation um, to differentiate it from the double dissociation. So dissociation is where there is always a disruption in one mental process and there isn't a single case where there was a disruption in the other mental process alone. A double dissociation is when the both the kinds of cases are found. In other words, there is a patient with a disruption to mental process A, but not B, and there is also another patient somewhere with a disruption to mental process B, but not A, suggesting that these two mental processes A and B are independent of each other most likely. So now again, keep in mind, if the patient record only indicates dissociation, that does not mean that there is no double dissociation. It's quite possible that the cases we have received so far have only been specific to disruption of mental process A. We might find later on a different kind of case where there is a deficiency, disruption to mental process B and not A. So we are still open to the possibility of a double dissociation in cases of dissociation, but not the other way around. Again, dissociation is disruption to only one all the time mental process. Double dissociation is disruption to one with a non-disruption to the other mental process, vice versa. Okay, a better way to understand this particular double dissociation and single dissociation difference is to think of the TV analogy. Now, just imagine, can you have only image in the TV and no sound? Well, it's possible, right? Can you have only sound but no image? Well, that's also possible. In other words, if you think of image and sound as mental processes, just as an analogy, and you're trying to see how related these two are, well, they are quite separable. Separable in the sense that you can have one without the other. You can have image without sound, and you can have sound without image, suggesting that these are doubly dissociated. In other words, they are well separable. One exists without the other. They are independent mental processes, most likely. That is double dissociation. Single dissociation is, can you have an image? Just think of an image on the TV without color. Well, black and white, right? You can have an image without color. Now think about this one. Can you have a color without image? Well, the moment you have some color, there is automatically some image attached to it, correct? So it's hard to think of color, or it's hard to think of color without image. What one is a part of the other, suggesting that these two, color and image, are not completely separable, because one is at a higher order than the other. The moment there is color, there is also image. So this is dissociation, when there is always a disruption to one mental process, image in this case, and not the other. So far, we've been talking about mental process A and B, not specifying what this could be. Now, we're going to use this example to kind of understand um, what we mean by these terms: association, dissociation, double dissociation. So, um, this individual is referred to as KC, um, and he had a motorcycle accident, and after the accident there were some changes noticed. For example, this particular patient KC, he can play chess, he can explain you the rules of chess, he can explain the concept of chess, the concept of the game, the logic of it, everything, but he doesn't remember ever playing this particular game. And if you watch this video, just type patient KC on YouTube and you'll find a lot of videos on this. And one of the videos, there is like 5 to 10 minute interview, and you'll realize that the individual has a really good understanding of some concepts. But when asked about his personal experiences of using those concepts, he would say he doesn't remember anything. For instance, he could explain in detail how to fix a flat car, but he doesn't remember a single instance of actually doing it. Now, think about it for a second. Can you identify two specific mental processes that are involved in this case? And which of these mental processes is disrupted? Again, the person can play chess or explain the rules of chess, but he doesn't remember the specific rules, sorry, specific instances of him playing. Which long term memory processes you think are involved? Well, take a pause here. In the next video, I'll reveal, but I would really like you to give a try. Well, you're absolutely right. He has a good knowledge of chess, the rules of chess, and everything. In other words, he has a really good semantic memory, but there is disruption of personal experiences, which is episodic memory. So, if all the cases are like KC patient, where there is always disruption to the semantic memory and not episodic memory, that would be an example of, that's right, dissociation. But if you see one patient like KC and maybe another patient, say CK, just as an example, where there is a disruption of semantic memory but not episodic memory, then that would be a double dissociation. I'm not saying there is one such case. This is just to give you an analogy. And definitely, this is an example of single dissociation. This brings us to the next outcome for this particular unit, which is the principles of neurocognition. And specifically, we're going to talk about, there are several principles, but we're specifically going to talk about the two most important principles for the purpose of this class. And those are contralaterality and hemispheric specialization. Now, I'm going to give you a general description of these two principles here, but when we I would strongly suggest you to watch the videos on brain. There are short videos, I think 4 or 5 short videos, which are like 4 minutes or 3 minutes each, that describes the forebrain, midbrain, and hindbrain part of the brain. And I talk more about this contralaterality and hemispheric specialization in split brain. So I strongly suggest you watch those videos. Okay, in this video though, we'll talk about contralaterality in general and hemispheric specialization in general and general definition of split brain patients. So contralaterality is the fact that um, one side of our body is usually controlled by the opposite side of the hemisphere. Again, this is described in detail in the forebrain lecture on brain. It's a short 5 minute video or 4 minute video. I strongly suggest you watch it. But contralaterality is the fact that one side of our body, your right side of the body, is controlled by your left hemisphere, and your left side of the body is controlled by your right hemisphere. Also, the sensations from the right side of the body are perceived or sensed or perceived by your left hemisphere, and the left side of the body is perceived by the right hemisphere. So this is contra is opposite, lateral is lateral. So your body is controlled by the opposite side of the body. It's not only controlled, but it's also sensed. And when I discuss about the sensory cortex and motor cortex in the cerebral cortex section of the brain video, it clearly explains what it means. So if I ask you to raise your right hand, well, if you did raise your right hand, that's because of the message you received from the left cerebral hemisphere, namely the motor cortex of it. So that's what contralaterality means. One side of the body is controlled by the other opposite hemisphere or sensed by the opposite hemisphere. Contralaterality in vision, though, is slightly different. It's not I'll talk about what exactly it means, but just to give you a general idea of this particular picture, just think of this is your left eye, this is your right eye, this is your left hemisphere occipital lobe visual cortex, and this is a right hemisphere occipital lobe visual cortex. And if you watch the videos on the brain, you'll learn that the occipital lobe is the seat of visual perception, and there are visual cortices in each of these visual hemispheres or the occipital lobes. Now, now what you see here is the visual field. Now it is represented here twice, but what it actually is that there is this distinction. So if you look straight ahead, and whatever you kind of see on your left side is your left visual field, whatever kind of you see on the right side is your right visual field, and put together is your entire visual field. Now the representation here is that the left side or left visual field is coded in blue, and the right hemisphere, right visual field, is coded in red. So left hemisphere, sorry, I keep saying hemisphere, left visual field is blue, right visual field is red. Okay, now it is represented here twice in relation to the eye, but hemisphere, the visual field itself, right, the left is blue and the right is red. Okay, now if you think about it, if you look at it, you'll observe that the red colored information from right hemisphere, it is going to both left eye and right eye. But eventually, the red information is going to the left hemisphere. Similarly, the blue information on the left side, right, is going to both left eye and right eye, but eventually it's going to the right hemisphere. In other words, there is contralaterality in vision also, but the contralaterality is not at the level of eye. In other words, the contralaterality here does not suggest that the information from left eye is going to the right hemisphere and right eye is going to the left hemisphere. No, information from both the eyes, right, go to both the hemispheres. You see that orange and blue, right? Orange and blue. So from each eye, the information goes to both hemispheres. So it's not at the level of the eye. The contralaterality is at the level of the visual field, such that the information in the left visual field goes to the right hemisphere, and the right visual field goes to the left hemisphere. And the reason for this is because of this weird crisscrossing of fibers. Now, in the sensation part, you'll learn that the optic nerve fibers from both the eyes, right, they crisscross in a way that half of the fibers go to the same side and the medial half go to the opposite side, leading to this crisscross of fibers called optic chiasm. Optic chiasm is this crisscross of fibers, and the crisscross is very interesting in that the lateral half, usually you call this part is a lateral, and the part that is the center is the medial part. So the lateral half of the bundles of fibers of the optic nerve go to the same side, but the medial half go to the opposite side, and because of this, you see that the contralaterality in vision is unique in the sense that it's not at the level of the eye, it's at the level of the visual field. So the coming to the second principle um, of cognitive science is the hemispheric specialization. Hemispheric specialization is the idea that your left and right hemispheres. Again, if you look at the structure of the brain, we'll talk about how hemispheres are formed. We'll also talk about what connects these two hemispheres. These are very short videos. I strongly suggest you read those, you watch those videos. Anyways, so the idea of hemispheric specialization, also called brain lateralization, is that the left and right hemispheres are specialized in functions. You might have heard about left brain is analytical, right brain is intuitive, and things like those. Well, there is some truth to it, but not in a sense that is actually used in colloquial terms. Like there is not actually left brain only about analytical thinking. It's not the right brain only about intuition or something. The idea here is, which is called hemispheric specialization. The idea here is that one hemisphere is more involved in some mental processes than the other, and that is hemispheric specialization. It doesn't mean that the other hemisphere is not involved in a given function. It's just that one specific hemisphere is more involved than the other. That's what hemispheric specialization means. Now, based on the same idea of hemispheric specialization, is this concept of split brain patient. Now, um, the whatever knowledge we have gained about hemispheric specialization, most of it has come from the split brain patient. Now, what are split brain patients? Well, for us to understand that, again, I strongly suggest you watch the video on forebrain, and I'll talk in detail about what the structure is in that video. But to just give you some basic idea, that the left hemisphere and the right hemisphere are completely disconnected. They don't talk to each other except for this particular band of fibers. So the left hemisphere and the right hemisphere are connected because of this band of fibers, and these fibers, this is called the corpus callosum. Again, watch the video. So corpus callosum is that band of fibers that connect each and every part of the left hemisphere, cerebral hemisphere, to the right hemisphere. If not for this bunch of fibers, these two hemispheres would never talk to one another. And and in some severe cases of epilepsy, actually, surgeons cut this particular bundle of fibers. The severe, they cut the bundle of fibers of corpus callosum. This is called colostomy. And the reason they do it is when there is severe epilepsy, which is uncontrolled electrical activity of a specific part of the brain. Let's assume there is a particular point on one hemisphere which is actually uncontrollable in its electrical activity, and this is the reason why a person is experiencing epilepsy. Um, is so these fibers actually carry these electrical activity information to the other part of the brain too, so the electrical activity becomes completely uncontrollable. In order to minimize the severe nature of the epilepsy, in some cases they cut the corpus callosum so that these electrical activities are not transferred to the opposite side, so at least you're reducing the spread by half, and most of the times it works. But this gives a unique situation where such patients who had this corpus callosum cut, their left and right hemispheres directly don't talk to one another. And then you have this uh, um, situation of split brain patient, and there are several experiments done on patients. And again, I talk more about this particular construct in the video about forebrain, so I strongly suggest you see that. So this is split brain patient. So what typically they do is they present information only to the one visual field and not the other visual field. And they don't let the individual move his head or her head so that they send information to one or the other hemisphere and try to see which one is more involved in, say, inferencing and things like those, and then gather more information about hemispheric specialization. So this brings us to the next outcome for this unit, which is the methodologies specific to neurocognition. Now, we can talk about at a very general level, basic level, we can talk about lesions, direct stimulation, and two of the imaging techniques, EEG and ERP. Lesion is when a specific part of the brain is either injured or damaged because of an illness, and you can connect that particular region with the disrupted mental process and make the connection and locate the mental process. So, lesion is one methodology. Um, Again, lesion could be because of an injury or because of an illness, um, damaging a specific part of the brain, and that way we can connect the structure to the function. So direct stimulation is when the cerebral cortex, the specific part is stimulated, and the evoked experience reported by the uh, patient is recorded. Now keep in mind that direct stimulation usually is not done for the purposes of direct stimulation, but when performing surgery to the brain with the open skull, this could be another um, thing that the physician might be interested in checking. That way they can connect the structure of the brain to the functions. And one person who has done a lot of research on this is the scientist by name Penfield. Um, and he would stimulate with a very minor uh, mini electrodes, or the electrodes with very little voltage, and then ask patients what they experience. So visual cortex experience, they would see something, the auditory they would hear something, and things like those, and that really helped later on for researchers to actually pinpoint different structures specific to different functions of the brain. Now, imaging techniques refer to several different ways, different methodologies, and the basic idea of this imaging technique is to take images of the brain um, under different um, circumstances, in the sense like either by through MRI or fMRI. So these are different imaging techniques. Again, the basic idea is taking pictures of the brain under, and then those images are captured and then examined either for the structural details or the functional aspects of the brain. And we'll talk about two such um, techniques. One is MRI, the other one is fMRI. Now, coming to the first imaging technique, MRI, which stands for magnetic resonance imaging. Now, magnetic resonance imaging is slightly similar to the basic fundamental idea is similar to an X-ray, um, but instead of X-ray, X-ray in the case of X-ray, um, we use X-ray to actually it passes through the body parts, and then you see the shadow, in the sense that the darker regions are related to the harder structures, and the lighter regions are related to the softer structures in the body. The same logic is used here, but instead of using X-rays, MRI uses the radio waves. Now, more important than using radio waves, which are not um, invasive in the sense that they're not as dangerous as the X-rays, so radio waves are safer than X-rays. But also, another underlying point about magnetic resonance imaging is that the most of our body is made of water, and water molecules have hydrogen and oxygen in them. And the hydrogen atom has a proton at the center. If you remember the protons and neutrons are a part of the nucleus, and the electrons surrounding the atom, structure of the atom. Remember that. So the hydrogen atoms have positively charged nucleus, or the proton at the center of the hydrogen atom, and so just imagine like each and every hydrogen atom in our body, and if you're specifically looking at the image of the brain, then each and every hydrogen atom in your each and every cell of your brain, it has magnetic properties, and would react to external magnets, just like metal pieces are magnetic, and you can align a dust of metal by putting a magnet. Have you ever played with that? You can move the magnets around, the small metal pieces around with the force of the magnet. Well, the same logic is used here, where the individual who's sent into this MRI machine is actually uh, um, would be surrounded by strong artificially generated magnetic field. This would realign the proton molecules, or the protons of the hydrogen atoms, or the molecules in the structures of the in the parts of the brain, and this alignment gives a specific signal. More importantly, when the radio waves are sent to the brain parts, then this aligned hydrogen molecules from the specific parts, the protons, they get knocked off, and when the radio waves are withdrawn, they realign to the magnetic field. There's a difference in the alignment. Some parts of the brain are harder, some parts are softer, Some parts the uh, hydrogen molecules are quicker to realign, the others are not. And so it all depends on the magnetic properties of the brain itself and the magnetic property of the external magnet. And with the help of the radio waves, we can figure out the specific structures involved of the brain. And you can look for the lesions, you can look for injuries, you can look for smaller details in the brain. It's more like, just imagine the pixel pictures. You can draw an entire image of a, say, the entire, I don't know, Eiffel Tower, let's say, by using pixels of information, right? Individually, the individual pixels have no real connection to the Eiffel Tower itself, but together they give the complete picture of the Eiffel Tower. Now, you can imagine these proton molecules also, or the protons also, as these pixels, and giving the complete picture of the brain. Now, again, MRI is not just used for brain. You can have MRI of any part of the body. MRIs give a better picture of the structures involved in the brain. And it's usually used to understand the structural changes or structures of the brain. So the fMRI is also called the functional MRI. Is different from MRI in the sense that MRI is used to take structural pictures of the brain. Functional MRI is used to understand the functions of the specific structures of the brain. So typically, an individual here would actually doing a task, and the fMRI would take pictures of the brain and localizing the parts that use more oxygen at any given point of time. The idea, the logic is that more oxygen, hence more blood flow to those parts that are used more when performing a task, opposed to the regions that are not used in the task. So it's like more blood flow. It's like taking the amount of blood in a specific task that you're doing. So if I give you a task and your frontal lobe is more involved, and there is more blood flow to the frontal lobe, you can tie connections to the frontal lobe, or specific parts of the frontal lobe, and the task you're doing, and the relationship between them, when compared to other parts of the brain where the blood flow is less. And again, it's more than blood flow in the sense that the blood flow is the increased blood flow is the idea of increased oxygen supply to the brain. And when there is increased oxygen supply to the brain, the RBCs carrying oxygen um, have, or any RBCs have hemoglobin in it, and the heme part, the iron part of the hemoglobin, acts differently. It has different magnetic properties when it is uh, carrying oxygen versus it's not carrying oxygen. And this difference in signals that the blood sends, called Bold signals, is what is captured by the machine. And that gives us a better understanding of which are the most active parts at any given point of a task. And we can figure out the body parts, sorry, the brain parts involved in specific tasks. Now, the electroencephalogram is another methodology. Here, um, what an electroencephalogram does is basically there are several electrodes um, that sense the electrical activities of the parts of the brain under the skull. Um, so it's more like, if you remember from your intro to psych discussions about brain waves, stages of brain activity and brain waves. Basically, EEG captures the brain waves, and which are basically represent the electrical activities of the brain at any given point of time. By capturing the electric activities of different parts of the brain at any given point of time, You can locate if there is any um, irregular activity. For instance, in patients with epilepsy, you can localize which part is actually causing this epilepsy, or which part is involved in epilepsy, which is basically uncontrolled electrical activity of a specific part of the brain, right? So the brain waves will be different for that part when compared to other parts of the brain, and you can locate the center of the epileptic seizures. So again, it's a simple process of measuring the electrical activity of the brain, and typically an individual is they use a cap and the many receiver electrodes that can detect the electrical changes in the brain, and then you pinpoint the electrical activity to the specific brain part. Event-related potential is pretty much same as the um, electroencephalogram, which measures and captures the electrical activity, except that it's more precise in a sense that instead of just measuring the electrical activities of the different brain parts, we're specifically interested in this case about a specific brain part involved in a specific task. So it's similar to EEG, but it's more localized and it's very specific to the task. What does it do? It measures it measures the electrical activity, but after a specific task. So an individual when presented with a stimulus, right, then you know a specific part of the brain and try to see what kind of momentary changes happen in electrical activity specific to the part that would give us more information about the mental process involved. So it measures momentary changes in electrical activities after a specific stimulus is given. So if I give you my name or a sentence to read, then have some anomaly in the sentence, and then observe what happens when you read that anomalous part of the sentence. And typically, there are several phenomena related to this, but there are two most commonly studied phenomena related to ERP are the N400 and P600. N400, N here refers to the negative or positive. Remember, you're measuring the electrical activity in terms of waves, right? And basically, you're looking at the amplitude of the waves, the length of the waves, right? The amplitude of the waves. And this is the baseline. And if it is above, it's positive. If it's below, it's sorry. If this is negative, this is positive. N400 is when you see a negative activity after 400 milliseconds of presentation of a stimulus. That's one phenomenon. The other phenomenon is P600, where you see a positive wave after 600 milliseconds of presentation of the stimulus. And we can talk about the phenomena related to N400 and P600 in the next slide. Well, I want you to read this sentence out loud. Okay, running out the door, Patty grabbed her jacket, basketball, gloves, cap, and a skyscraper. Well, you're right. This is a meaningless sentence, right? A sentence like this is typically referred to as a semantically anomalous sentence. The content of the sentence doesn't make sense. Structurally, syntactically, it's an accurate sentence, but meaning-wise, it doesn't make sense. It's more like the colorless green ideas slip furiously from the language topic we discussed. Okay, so in an ERP study, event related potential study, participants were presented with sentences like this. So, researchers would look at what happens after the reader reads this last sentence. Okay, and usually semantic anomalies are associated with N400s. What are N400s? N400s are these negative changes in the electrical activities a sudden change in the electrical activity in the negative side after 400 milliseconds has elapsed. So after 400 milliseconds of reading, after 400 milliseconds of reading of skyscraper, typically you see a negative wave. That's why it's called n negative 400 after 400 milliseconds. So semantic anomaly is associated typically with n 400. Another task, read these two sentences. Participants typically are presented with sentences like John told the man to leave, or John hoped the man to leave. Now, if you look at it, this is grammatically or syntactically correct, but this is not syntactically correct. This is a syntactically anomalous sentence. This is called syntactic anomaly. So you can't say John hoped the man would to leave. You can say John hoped the man would leave or something along those lines. So this is syntactically anomalous sentence, and typically this is associated with a positive 600 P600 phenomenon, where participants, when compared to syntactically normal sentence, in syntactically anomalous sentences, you see a P600. In other words, after 600 milliseconds of reading the sentence, participants would show a positive sudden change in the electrical activity of a specific part of the brain. So syntactic anomaly is associated with P600 and semantic anomaly is associated with N400. So these different electric patterns will give us information about the mental process involved in reading syntactically versus semantically anomalous uh, uh, sentences. Well, this brings us to the last outcome for this video. Again, in this video, I'm going to give you only a very brief discussion on the nervous system. But there are other videos that I want you to look at to get a complete picture of the structures involved in nervous system. Um, when we say nervous system, we're talking about the neurons, and we're talking about the classification nervous system. So I strongly suggest you watch. These are very short videos, 4 or 5 minutes each, and there's several though. So find time and watch those. That will really enhance your understanding of the deeper discussions we'll eventually make and the connections you can make by watching these videos. So, some basics of nervous system. Well, we all know a nervous system are made of neurons, and not only neurons, but there are other supporting cells called glial cells. Neurons are the special cells um, that have the capacity to actually receive, transfer, and process information. Typically, a neuron has a structure called the cell body or cyton, or then one large extension called the axon. There are several smaller extensions called dendrite, and usually dendrites bring information to the cyton, and the axons take information away from the cyton. And also, again, watch the video. There is a lot more information in there, other videos. And axons of some neurons are covered by these extensions of the glial cells to form a covering called myelin sheath, which insulates these axons. So when the information passes is received by the dendrites, is brought to the cell body, right? Then it initiates some changes, and these changes are called action potentials. These action potentials will go from one node of Ranvier to another. Nodes of Ranviers are places on the axon without the myelin sheath. Eventually, these action potentials reach the ends of the terminal buttons, and from there it goes to the next neuron by passing through a space called synapse. Again, this is basic information. There's no need to go over this. I still want you to bigger kind of give a quick review. You can check your intro to psych books, or you can watch the videos I've posted. Well, to give you a bigger picture of the nervous system, again, watch the videos. It has a lot of information. There are handouts too. Try to fill in the blanks before you watch this part. So the nervous system itself is made of neurons, which are specialized cells, and these neurons form structures, right? And these structures, you can classify the structure involved in the nervous system into central nervous system and peripheral nervous system. Central nervous system comprises of the structures that are centrally located in our body and play a central role, of course. One is the brain, and the other one is the spinal cord. The way these are written in this way is to kind of show some analogy to the actual shape of the brain and spinal cord. Um, so, central nervous system, brain, and spinal cord. Then there is um, then the peripheral nervous system, it's brain, spinal cord, and the nerves that come out of and go to the central nervous system. Those form the peripheral nervous system. Peripheral nerves connect the central nervous system, that is brain and spinal cord, with the rest of the body. And the two types of peripheral nervous system. The peripheral nervous system, the peripheral nervous system, um, one which is somatosensory nervous system, it connects the brain and spinal cord. It connects the brain and spinal cord um, with the sense organs and voluntary muscles. Then the peripheral nervous system of the second kind, called autonomic nervous system, it connects the other body parts with the brain and spinal cord. And there are two kinds of autonomic nervous system, sympathetic and parasympathetic. Again, I strongly suggest you watch the other video because I can't do justice to this part of the lecture in this one slide. So there's another video that you can watch to get a better picture. Thank you very much. This would bring us to the end of this particular topic of 4.2 nervous system. And we're done with this unit. So stay tuned. Have fun learning. Thank you.